The overall goal of this procedure is to track the retrograde axonal transport of brain-derived neurotrophic factor, or BDNF, by live imaging. This is accomplished by first assembling microfluidic chambers on polylysine-coated cover slips. In the second step, dissociated rat E18 hippocampal neurons are dissected and plated in the cell body compartment of the microfluidic chamber. Next, the neurons are cultured to allow extension of the axons across the microgrooves into the axonal compartment of the chamber. In the final step, quantum dot-labeled BDNF is added to the axon compartment. Ultimately, fluorescence microscopy can be used to assess BDNF movement within the axons of the hippocampal neuron.
The main advantage of this technique over existing methods like BDNF-EGIP or BDNF-M cherry labeling is that labeling BDNF with quantum dot allows the live imaging of a single BDNF dimers within the axons. Demonstrating the procedure will be Xiaobei Zhao, a postdoc from my laboratory.
Begin by hand washing the microfluidic chambers in 1% alkanox, followed by 3 30-minute rinses in milli-Q water. Then after hand washing the chambers in 70% ethanol, lay them out to dry on parafilm in a laminar flow hood. Sterilize both sides of the chambers under UV light for